Swayam Prabha. Digital India. Educated India.
Okay, so molecular biologists and biophysicists for a long time have been interested to analyze every possible interaction of RNA polymerase with the target DNA sequence. And they have identified a large number of sequences that are present in the promoter site that interact with RNA polymerase. So there's this -10 region that interacts with the sigma. Then we have alpha CTD, then we have one subunit, we have this other subunit, and then what they have made have been to kind of analyze one by one all the possible sequences that touch physically the polymerase, and what they have made have been to make a large collection of variants of each of these promoters. Okay, so as it is in principle difficult to calculate from very, very first principles what is going to be the interactions between this protein and the promoter, what they say is, well, let's generate thousands and millions of promoters with different sequences. And then later, we apply machine learning to identify and to associate different sequences with the promoters that we have. So these are the regions that interact with the RNA polymerase. And if you see here, what they make is that they make a combinatorial library of DNA sequences, and they put a reporter downstream of that, and then they see what is the consequence of having different sequences in the final outcome of transcription. Okay, so in this region you have different constraints. Then you have here all possible sequences, etc. And you generate a very, very, very large collection of promoters, and then you just make the experiment one by one. And then you generate a correlation or a kind of function between the predicted interactions, the physics of the predicted interactions between the RNA polymerase and the promoter, and then the actual transcription rate that you measure with a reporter. Okay, so in practical terms, if you are interested, then you can sit in front of your computer and then without making any experiment. Yes?
What was the sample size of the previous experiment?
Excuse me?
Delta G total. So basically, how many samples are there?
Well, maybe in the range of various thousands. Yeah, because this is made with a robot. I mean, the experimental setup is made with a robot. It's not a human that makes the experiment.
Okay, but Professor, see, there are so many segments of.
Yeah, I mean, you don't cover the entire combinatorial universe. I mean, that's very clear. You know. If you have a subsample, but still the subsample is enough to feed the data to a machine learning platform, okay? And then at the end, the computer makes a correlation between different types of interactions here and promoter output, okay?
Is there a science behind the sample? Like you do an initial number of samples and get, depending on the result that you get, you refine the way you do sample.
Well, you can do that. I'm not sure that they did that, but certainly, this laboratory, this group, is famous for, blowing up the number of samples and learning from, this machine learning process. I mean, you take the samples that you have, you take only a part of the fraction, you make a prediction, and then you confront that with the sample that you don't, that you have not entered into learning machine, and then you adjust the prediction and then go on and go on and go on. So this is the thing. Okay?
So what is the name of this?
Well, I will give you a name later. It's this guy called Howard Salis. Well, as we have stopped commenting papers, I did not include these papers in the list of reading, but I will be happy. I have the papers. I can send it to you. Is Howard?
Please.
Salis. So you put in internet Salis calculator you get all the papers instantly. Yeah? Because he has developed various computational platforms, not only to predict promoters, but also to predict ribosomes. I will discuss it in a moment, okay? So, you can sit in front of a computer and then decide what is exactly the composition of a sequence to give you a given promoter output within a given range, okay? You have a kind of linear relation, more or less, in such a way that you can have, in principle, a good estimation of what transcription you are going to have under given sequences. And by the same token, you can ask the system to give you a sequence that, if converted or entered into a promoter, will give you a given level of transcription. Okay, so that will be the theory. The same story goes for calculation of ribosome strength. So ribosome strength means that the ribosomes get very, very strongly into the ribosome binding site, and therefore transcription and therefore translation goes much, much better. But it's not only the binding of the ribosome to the ribosome binding site. It's also obstacles that the ribosome may find in its way through the open reading frame, through the gene. So again, Salis has produced a number of calculators on what would be not only the best ribosomal binding sites, but also what, sequence features you may want to eliminate to make sure that the ribosome, once it enters into the right open reading frame, it proceeds smoothly till the end of the gene, okay? So I insist that this is an entirely computational approach, no experiments whatsoever, or experiments made by a robot just to generate data to feed a machine learning platform, okay? Okay, then, well, you can then play with the data and make very, very variants and use part of the data to, as a kind of training set, and then you have a test set, and then you see whether they do to fit. If they fit, great. If they don't, you keep on adding more data to the learning platform and so on and so on. And at the end, you have a kind of pipeline through which you can combine both transcription and translation and predict the outcome of the whole gene expression flow, okay? So that's great, and in principle, and this, this type of work obviously has been very well appreciated by engineers for the most part that don't like to make experiments in the laboratory and to get the hands dirty for the most part, is that you sit in front of the computer and without having any knowledge about molecular biology, about cells, about you name it, then they can write a DNA sequence that, if put in a biological platform for expression, in principle, it should work as predicted. That's just fantastic, okay? However, things in reality are a bit more complicated, in the sense that in many cases, in synthetic biology, what you want to do is not just to optimize one given pathway with a promoter, gene or a set of genes, and a few ribosome binding sites in front of them. In many cases, what you have is the challenge to make a complex biological, say, item where you have to optimize not one or two or three things. You have to optimize many, many things. For instance, I'll show you one example coming from one project that is going on in our experiment, and is to have bacteria, in particular Pseudomonas putida. I'm sorry to make so much propaganda about Pseudomonas putida, but it's the system we work with. But what I say can be extended to any other bacterium. What we want is to have a bacterium that is able to degrade cellulose, for instance, okay? Okay, so what is at stake when you want to make such a catalyst? Well, it's not just that you have to have a pathway for degradation of the say, residues of the cellulose. You need to have first a cellulosome or components of a cellulosome. And remember that in previous days I mentioned some experiments that enable to display on the surface of the cells these complexes that convert cellulose into glucose, but then it has to be entered into the metabolism, and it can be, ideally, it has to be combined with other features here. So you have the problem of channeling the results of these reactions to the metabolism. You have the channel of debugging the interplay between the chassis and the implant. Remember this terminology that we have discussed. You have the issue of sensing the flow of carbon. You have to manage the stress. You have to do something also to ease the downstream processing. So under many conditions, or I mean, in most circumstances in biotechnology, the issue is not just to optimize one bit of the whole thing, but to optimize the whole thing. And it is frequently the case that you don't have all parameters that you need. You may have some of them, but you don't have all of them, because there are many, many, many parameters involved in optimization. So the challenge then turns into finding a solution in a solution space. So you may determine what you want, what would be the optimal situation that you may want to have, and then you have to survey, you have to navigate through a solution space, and then if you are lucky, you may end up with an optimal design. And therefore, the problem of optimization of a synthetic biology agent for biotechnological applications turns into the engineering problem of multi-objective optimality. So this multi-objective optimality, and this is a slide that I showed you before, is connected to the fact that it's not enough that one process happens independently. It has to fit some parameters that ensure that the inputs and the outputs of every step are compatible with the next steps, right? So, well, this is simplified. You may have a nice igloo, you may have a bird here, but if the dimensions do not fit, then you are lost. So you cannot really do much, okay? Okay, so engineers and mathematicians for a long time have played with this concept of Pareto surfaces that have to do with multi-objective optimization. And in biology, there is a direct connection between the mathematical concept of Pareto surfaces and what is called fitness landscapes. So fitness landscapes is this symbolic representation of how cells do under various circumstances and what are the consequences of adding more or less genes to a given biological chassis, okay? So depending on the circumstances, then you may have more or less optimal solutions, and in general, the optimal solution is the composition from partial Pareto surfaces, okay? So, I mean, in a simple Pareto surface, you have performance, cost, and reliability. These Pareto surfaces are popular in economics in general, but, you know, they can be transplanted easily to the biological world because, as I said, these fitness landscapes are the consequence of having to fit many different objectives at the same time, okay? So how can you do that? And how engineering and technology has solved the problem? How do you deal with a challenge where you have very clear what you want to construct, but you don't know all the parameters. However, you want to do the right thing. So how do you handle that? So let me show you a good example of how technology can solve the problem of constructing an object that is very complex without knowing all the parameters. Well, I show you here a famous building in Spain. There is a cathedral. It's a cathedral. It's called Sagrada Familia. But you know, if you go to Barcelona at some point of your life, this is one of the buildings that you have to see. So this building started to be built in the mid-19th century. By that time, there was no computation, no advanced physics, no advanced mathematical calculation, no advanced material science, and still, if you look at the structure of the cathedral, it's very interesting and very complex, okay? So you have, you get inside, then you see a whole number of arches that are connected in very, say, intricate patterns here. And well, you may notice that you don't have like straight lines necessarily. You have like straight lines, but at some point they bend a little bit, and then they bend a little bit, and so on. And well, you can spend hours looking at the building, and the interior is really spectacular in the sense that it covers a large space, a large volume of three-dimensional, say, architecture. And well, you wonder how people of that time were able to construct such a complex buildings without knowing the mathematics and the tools that are now common in engineering and in modern architecture. Okay, so the solution comes from the ideas of this guy here called Gaudí. That is one of the most, say, famous architects in history. And he had this brilliant idea. He said, well, I cannot calculate all the parameters. I cannot make a building with predicting everything, because there are many, many aspects that I don't know. So, but still I want to make the building. So what he did was to make models of strings and weights in such a way that he said, well, if I want to have a structure of this type, let's put weights in strategic positions, and then as every weight will represent a kind of parameter that otherwise I will have to adjust, okay? Like this, no? If you put a weight here, you know that the angle to the next one will be this one, this one, this one. And well, the solution to the problem of what geometrical figure you get when you put weights here, here, and there can be calculated or not, but the gravity will give you instantly a solution, okay? So this is the type of things that he did. So you make this complex, say, model where every weight that you put here would be equivalent to an individual parameter, but keep in mind that as everything is connected to everything, every value of one parameter will be influencing the behavior of the nearby components. And this is exactly what happens in the biological system. You can have here a very nice promoter, but you know, as it is surrounded by many other things, then at the end it may be different. So what the guy did was to make these models, and then what they did was to turn them around. And by turning them around, what you have now is a perfect solution to the problem that you had before, because just by applying gravity to what the model, then you instantly have the solution to all the angles, all the connections, and all the, say, peculiarities that this complex model requires for construction, even if you know zero about the mathematics and the simulations and the properties that otherwise will have to be calculated from first principle. So that means that optimality in many cases, or in some cases, can be obtained without any number, just by leveraging what nature gives to us. So a kind of simple example will be the calculation of a catenary. Okay, so catenaries are the curves that results from hanging something between two pillars affected by gravity. And this is important because civil engineers know that to build a bridge, you have to make a catenary angle. Okay, so the catenary angle is exactly what ensures that the bridge will be solid and will stay there forever. Now, engineers and physicists have calculated catenaries for a long time. Okay. And you can make a mathematical model, you can calculate the tension, you can put the span. I mean, this would be how to produce a catenary from first principles, from mathematical principles, from physical principles. But if you don't do that, how can you build a bridge? Well, very easy. All you have to do is to figure out the starting point and the final point of the bridge. Then you just apply gravity in a model, and instantly the physics, I mean just pure physics, will give you the solution to a problem that could be calculated from first principle, but the solution of which can also be obtained through a completely different procedure that does not involve mathematical calculations. This applies not only to one catenary, but it can be, a complex catenary. And similarly to the building I showed you before, it can even be applied to a super big complex thing. So the take-home lesson is that in complex systems, you can identify optimality without the need to know exactly all the parameters. So look, then we have here like two approaches. One of them is that you have all these calculators for promoters, for ribosomal sites and so on. And then that's great for the engineer because if you know all the parameters, no problem. Then you can do the construct, and the construct will work as you expect. But if you don't know the parameters, and that happens all the time in biology, or you know only some parameters, but not all of them, then the only way that we have to solve the complexity design problem is to rely on a different principle. So imagine that in this case, you have this construct that you can do with a promoter, with a regulator. You want to convert this product into a system and so on. So how do you, what would be the equivalence of this situation that you have here to this other equivalence that you have there? Okay, so this would be the kind of similarity. So one can speculate that evolution enables the gravitation of a complex biological system towards optimality, to what engineers call optimality. So how can you do that? Well, imagine that you have here like 6 activities, 6 genes that you want to optimize in terms of their expression. So you start by connecting them somehow, but then at some point what you do is that you apply to the body that is formed by the different components some type of pressure that would be equivalent to the say gravity that we have in these complex buildings, and then, well, the gravity itself will give you the solution to the structural problem in the cathedral. Evolution will give you the solution to the optimization problem in a biological setup. Okay. So this idea of solving a complex problem through physical means is called heterotic computation. So it's you are used to computation, you know, with computers and so that are based in Turing's principles and other, say, principles. This is a different type of computation. Okay, so this computation is one in which you embody the problem in a material object, and then you let it to undergo fluctuations, and then you have to apply some type of physical force, and the outcome is a physical measure. It's not a material. It's not a number. It's a material object that you may not even know exactly the parameters or the numerical parameters associated to it. I'll show you an example. Let me see if I can. Okay, do you have a string somehow? Can you borrow your? Yeah. Okay. Okay, thank you. I will take it with care, don't worry. Okay, so imagine that you have the mathematical problem, or the problem, you have this thing here, and you want to calculate what is the middle point of this object, okay? Then, no problem. I can go through a mathematical procedure. I take a meter, I measure from here to here, and will give me a number, and the number might be 150 centimeters. I don't know if it is like that, but more or less, right? And then I write 150, and then as I want to know the middle point, I make a mathematical operation that is a division, and then I divide between 2, and then the mathematical operation will tell me that the middle point is 75 centimeters. That's a numerical solution. Or I can just take this string, go to this string, and bingo, the solution is this one. You know, no mathematics, no nothing, but it's the same solution. It's exactly the same solution. Okay, thank you. Beautiful full art, by the way. Okay, great. So this is at large scale what is called heterotic computation. That you make computation, you pursue optimality, but without calculations from first principles. Okay. Obviously, with the time, if you have more and more and more parameters in a system, then you don't have to make so much heterotic computation, so much evolution, because the parameters will suffice for developing or for solving a complexity problem. But if you don't get to the point that you have so many parameters, then you have to rely in this kind of unusual ways of computation. So in summary, one way or the other, whether through direct, rational, and completely directed engineering, or by this type of unusual approach, then the endeavor is about finding attractors by fluctuating the object in a solution space. Okay, So, and this principle that I just mentioned is not only for biology. It has been applied back and forth between biological systems and evolutionary systems. Engineering systems and biological systems. There is a famous case that I want to share with you. That is the famous case of the ST5 antenna made by NASA to capture cosmic radiations in the exterior of their spacecrafts. Okay, so the engineering problem is how can I optimize the shape of the antenna to really make sure that I capture all the signals that I'm interested in. Okay, an engineer, I mean, to answer that question, you have so many parameters involved that you cannot calculate that from first principles. So what NASA engineers did was to start with an evolutionary algorithm in such a way that little by little, you know, just play with one say segment of wire, and then it started to move around, and then depending on the movement, then it was different types of radiation was captured, and then at some point, if the system started to be beneficial in respect to the previous conformation, it was the starting point for making another one, and then that was the basis for making another one, and another one, and another one. And at the end, the result was these antenna designs that were absolutely optimal, absolutely fantastic, but that work, no one knows very well how. Simply because the parameters that are embodied in the construct, in the design, are so high if they had to be made by an engineer that they're impossible to handle. Okay. So, however, we had these strange, say, solutions that work very, very well, and the structure of which and the architecture of which was not calculated by an engineer. It was just the result of an evolutionary algorithm. Okay, so this is one of the cases. There are many others where this evolved antenna that is designed by a computer using a program that uses evolution, not a program that engineers use to handle different types of parameters. Okay, so this is something that to me is very inspiring, in the sense that even without knowing all the parameters of a complex system or a complex challenge, still we can do something. Okay. Therefore, to build a functional object, whether it's a biological construct or an engineered construct, like engineers do, then there is a gradient between what to do when you have when we know many parameters or you know very few parameters. So if you don't know parameters or very few parameters, the only choice that you have to optimize a functional object is through evolution. If you know all the parameters, then you can go entirely rational. Okay? But what happens in many cases is that we are in between, and both in engineering and in synthetic biology, sometimes we know some parameters, but we don't know all of them. And therefore, the parameters that we don't know have to be filled through some type of evolutionary approaches. Okay? So then the situation is that when you have a pathway and you want to optimize the pathway, you apply this concept of heterotic computing, and then you let the system to fluctuate by applying some type of pressure, and then at the end, the system, if you are patient enough, will give you the solution to the problem. So perhaps some constructs are easy to predict with these tools that I told you before, but in other cases, in most cases, as a matter of fact, these constructs are difficult to optimize through first principle. And then necessarily, you like it or not, the final step has to be always some type of evolutionary approach. Okay, okay, so then that takes us to a kind of grand view of how you connect technology with evolution. And well, there's this famous quotation by Arthur C. Clarke saying that any sufficiently advanced technology is indistinguishable from magic. You know, so if we show a mobile phone to a Roman or to one of our ancestors, for them that will be completely magic, right? So, and you know, but my proposition is that any sufficiently developed engineering is indistinguishable from evolution. Okay, so sometimes these kind of controversies between engineering and evolution, in my opinion, are not well founded, because at the end, everything is about finding solutions in a solution space. And you can find the same solution through direct engineering, through rational engineering, or through this type of heterotic computation that is based in evolutionary phenomena. Okay? Okay, well, you can see that very clearly, that technology and evolution, in many cases, converge. It's not only that you have convergent evolution between biological systems, it's also you have convergent evolution between biological systems and human-made systems, right? So why? Because, to fly, I mean, how many solutions are in this solution space for flying? Well, there are just a few solutions. You don't have an infinite number of solutions, and therefore it makes sense that these solutions can be found either by pure, say, evolutionary approaches or by direct rational engineering. And at the end, they converge to the same point, because these solutions are like attractors in a solution space. Another example that I like very much, and to me is very surprising, is this idea, right? So sometimes you say, well, birds have had wings, and therefore engineers look at the wings and eventually develop the wings for planes, right? So they saw how nature worked, and then the engineers imitated what nature did. And therefore, the biology discovered the solution before engineers discovered it. But sometimes it's just the contrary. Sometimes engineers think that they have discovered a solution to a problem, and then later it turns out that the solution that they have found is also found in natural cases. Okay? So one typical case, one typical engineering solution to the problem of coordinating the movement of two wheels is to have them dentated, and then to have a mechanism through which you have a driver wheel and a driven wheel, and then the motion of all of them gets assembled and gets coordinated because of that. Okay, so this it has always thought that this is a product of human, say, creativity. Okay, well, recently, a few years ago, it was discovered that the mechanism through which some grasshoppers coordinate the movement of the very strong movement of the two legs to jump is exactly this type of mechanism. You have like two wheels that interact together, and by doing that, they can really coordinate the movements in a very, very accurate fashion here. So it's, in my opinion, a very spectacular case where you see how the engineering solution to the problem is identical to the biological solution to the same problem. Okay? In one case, rationally devised, in another case, evolved. Because in the solution space, you have an attractor for this particular problem, and the attractor maybe just one. There are not many others, and therefore it has been found through this procedure. And therefore, and this is something I showed before, to build a functional concept, you have a functional object, you have to be somewhere in between. Okay, so far I gave you a nice kind of theory on how it works, how nature solves the problem of optimizing complex systems, but how does this happen in reality? Okay, so do we have? Can I convince you that this is something that nature makes on a regular basis? That what you do is that the system explores a solution space, and then it finds some say attractors to which the evolution of the system gravitates. Okay, so I'll show you a few examples of that. Okay, not a few, just a few. Okay, obviously, in my laboratory, always interested in this type of situations. We have for some time worked on a very, very specific problem, and it's the problem of degradation, biodegradation of this compound here that is called 2,4-dinitrotoluene. Okay, so 2,4-dinitrotoluene is an interesting molecule because it's present as a residue of explosives production. Also it is produced during the synthesis of some foams, and it's a very, very toxic compound. But what happens is that this is a typical so-called xenobiotic compound. Xenobiotic means that this is a compound that has never been in nature before synthetic chemists put it there. Okay, and therefore a bacteria, the entire environmental microbiota, had still very little time to develop the ability to degrade it completely, right? So you can find bacteria that degrade it a little bit, but it's clear that they have not yet solved, have not yet optimized the problem of solving that. However, there are some bacteria out there, for instance, this bacterium called Bucholderia cepacia, that can do something, can degrade a bit this compound here, but is not yet optimal, very, very clearly, okay? So this bacterium was isolated in the US and so on, and it, when you look at the genes that are involved in this partial or incomplete biodegradation, then you find that, well, it can be degraded a bit, but not a lot, okay? So you can consider that this bacterium is in the way to optimize a pathway for degradation of this compound, but the pathway is not yet there, okay? So the pathway, and I don't pretend that you remember all the steps, involves a typical biodegradation pathway for aromatic compounds. That involves mostly the first step, the dioxygenation of the ring for producing a catechol, and then the catechol continues and continues. I have to say, as a kind of side comment, that biodegradation of toxic compounds systematically starts by oxygenation of the compound, and if it is an aromatic compound, normally you have a dioxygenation. And the reason for that, and it has a very strong logic, is that the aromatic rings are very stable, and if you introduce two adjacent OHs at the sides of the ring, then you destabilize that part of the molecule that then becomes more amenable to further biodegradation. So in general, the most important step in any biodegradation pathway is the first step. Once you form a catechol, the rest is piece of cake, okay? So it's always the question of how you engineer that particular, or how you evolve, or how bacteria invent a pathway or an enzyme able to dioxygenate the aromatic ring. Okay, so if you take a look to the origins of that gene, and it's also quite interesting, what you realize is that the pathway for degradation of 2,4-DNT have not come out of the blue. It is the result of, or it has as an ancestor, pathways known for degradation of naphthalene. So this is also typical in the world of bioremediation and biodegradation, that sometimes some existing pathways for natural compounds, and naphthalene is a natural compound that is in the petroleum, and therefore bacteria in this case had plenty of time to evolve good pathways for the biodegradation. But it happens in this case that the genes, the DNA sequences, have been recycled by this other bacterium, making some changes here and there, and the percentages are here, to degrade 2,4-DNT instead of degrading naphthalene. But there's something very funny when you expose this bacteria to naphthalene and to 2,4-DNT. Naphthalene is what you may call the old substrate, and 2,4-DNT is the new substrate. So what happens is that if you expose this bacterium, Bucholderia cepacia, to succinate, nothing happens, of course. If you expose it to either naphthalene or 2,4-DNT, the compound is degraded here, okay? However, if the genes are deleted, then obviously the compound is not degraded. But look at the survival of the cells. So this bacterium here is able to degrade naphthalene, but the population dies in the process, right? So it's like, you know, you started here, the bacterium can eat this compound, but the population decreases. So it's not a very good business, right? So like you have a victory in terms that you can eat the toxic compound, but most of the population dies. Is what is called a Pyrrhic victory. So Pyrrhic victory is when you make a big win in a battle, but the losses are so immense that you wonder why you started the war to start with, okay? And yes?
So in the delta DNT, which means that the genes for synthesizing or metabolizing DNT is not there. So you are going, and when you are giving 2,4-DNT, what is the, so the cell cannot metabolize DNT, second graph. So what is the carbon source, or what is the source of this nutrition?
Okay, well, in this case, I mean, the cells grow either in succinate or in naphthalene or in 2,4-DNT, okay, in both cases. So when they are in 2,4-DNT, then the population drops.
I get that, sir. So the second graph.
Yeah.
The second graph. So in the so basically the gray line is the one where only the carbon source is just DNT, correct?
Well,
when cells do not have the genes to metabolize DNT, how can they grow?
okay, so in this case, and this goes into the detail of the experiment, what we do is that we maintain the cells on succinate, and then we add either naphthalene or 2,4-DNT. Okay, good question. Okay, so why cells are so, damaged by this? Well, the reason for that, and there's a chemical reason for that, is that the first step in the degradation involves a chemical reaction that, as a result of the reaction, generates reactive oxygen species, okay? And reactive oxygen species are toxic, but at the same time they are mutagenic, okay? And therefore, what happens is that when you expose cells to 4-DNT, then, or to the older substrate, then cells might die, or a large percentage of the population may die, but at the same time they generate diversity, they generate mutations, and therefore the system starts surveying for, in this solution space, to find a given solution to the problem of degradation. So if you don't have this is a way of measuring the frequency of mutations by looking at one marker, that is resistance to rifampicin. And what happens is that when cells grow in these substrates, then cells not only degrade the substrates more or less, but they also generate diversity. They start mutating, and at the end, the surviving population that you have is hyper-diverse. And when you are hyper-diverse, chances are that you will find a solution to your problem, okay? So this can be abstracted in this way, in such a way that faulty reactions, reactions, stress caused by the fact that the pathway is not yet completely optimized, causes reactive oxygen species, and the reactive oxygen species create mutations, and the mutations create novelty. And therefore, under metabolically generated reactive oxygen species, the survival may decrease, but the diversity will increase a lot. And therefore, there is a kind of window of opportunity between stress, mutagenesis, and lethality, where you can really create very new things, and you have a kind of productive diversification. So that means that bacteria have developed, or bacteria and general biological systems, a kind of trick to self-adapt to the medium or to the environmental challenge on the basis of creating stress and converting stress into a source of variability. So this is what we may call the Redekos metabolic innovation cycle. So if you consider only the classical central dogma, you have DNA, RNA, proteins, period. But there's no way that you can go from proteins back to DNA. Well, that's partly, that's true in part, but it's not completely true, in the sense that now proteins produce metabolism, and metabolism can go wrong, and the cells, as the consequence of this wrongdoing metabolically, can create ROS, and ROS in turn will generate a situation where the DNA has to mutate here and there until the entire setup is able to solve a solution to the problem, okay? So this is the way that, in many cases, bacteria use in the environment to solve these new challenges. So you are faced with a new nutrient. The new nutrient is not palatable, but it's toxic, and then this toxicity and this stress generates, in turn, some stress that is converted into toxicity, and then part of the population dies, but the other population hyper-diversifies, and as a consequence of that hyper-diversification, you have a quick evolution towards solving a problem. So, well, this is how nature does it. So, question is, can we do that in the laboratory? Can we start with a situation where you have a dispersion of points, but then after a while you can, find a point here or there that are solutions to your particular problem? Okay, so that will be the next question to address. Obviously, well, not obviously, but the way to reproduce in the laboratory what nature has invented already is to apply different techniques, or what is called adaptive laboratory evolution. So adaptive laboratory evolution is no more and no less than trying to accelerate in the laboratory happenings that occur in nature under normal circumstances, but under more controlled conditions, and mostly directing the evolution of the population in just the direction that we are interested. Okay? Well, I'll show you just one setup that we have in the laboratory, run by this gentleman here called David. And then you can run these ale experiments from very, very simple to super complex. Okay? So in this case, for instance, simple is that every day you change the culture from one flash to the other, and next day, and next day. You can do it manually. But in this case, what he did, or we did, was to automate a reactor that is constantly having a culture that is set for a particular objective. And the particular objective is, in this case, to make a connection with what I told you before, degradation of 2,4-D-nitrotoluene. Okay? So we can make these say changes in a robot, or with this continuous system that has sensors in different places to monitor the levels of intermediates that we accumulate or that we get rid of. Okay? And in this way, you can really control well the evolution. So you can do that in a kind of trivial way with just manually and so on. But also, as I said, you can have different sensors, and the sensors will give you the levels of different metabolites, and then you can select conditions that will favor the accumulation of some metabolites or not metabolites, and then check for growth. I mean, today you have different available adaptive laboratory evolution setups that will really multiply by many orders of magnitude the spontaneous power that bacteria may have to evolve under other conditions. Okay? So this is just one experiment, for instance, in which we started with one with accumulation. I mean, we monitor accumulation of 3 intermediates, and then in different conditions, then you see that the accumulation goes in different directions and so, and at the end you can select the one in which you have a prediction that having one of the metabolites high, the other intermediate, and the other low will be the best starting point for another round of evolution. Okay? So at the end, as a consequence of this type of manipulations in the laboratory, and after 1 month of evolution, we were able to create a strain of Pseudomonas putida able to degrade altogether to 4-DNT. Okay? So this is the type of case where you could think on optimizing the bias, playing with the promoters, playing with the ribosomal Sandel Garnos, all these things. But to optimize a degradation process, you have to nest that particular device in the existing biochemical network of the host. If you want to do that in vivo, there are so many parameters involved that you really cannot do it. Okay? However, by making this experimental evolution, we were able to have a strain that eventually, as you see here, can produce biomass out of added to 4-DNT. So that was something that was very, very nice. So these days, given the realization that one cannot really optimize systems exclusively based on basic on first principles, then there are more and more propositions about having this ALE or these adaptive laboratory experiments setups applicable to many other things. This is another way that this is a chemical engineer that has developed this kind of blueprint here, and then you can try to set up a very good system for this type of experiments. For instance, this particular device as a detail, and you are getting here more into chemical engineering, not so much in biology. One of the problems when you make these evolutionary experiments in the laboratory in a continuous fashion is that bacteria tend to stick to the surfaces, and then you may have bacteria growing in a flask, and then you can select for some trait, but then after a while bacteria will stick to the walls, and that will kind of screw up your experiment. So in this case, for instance, David developed a concept, and is to have the reactors flush every 24 hours with sodium hydroxide, and by doing that, then we ensure that any remaining cell stuck to the surface of the reactor will be eliminated. Obviously, this was again washed and everything, and then you can maintain this operation for months and months and months, and still maintain a degree of sterility and avoid the formation of biofilms. Okay, so there's a kind of branch of synthetic biology that has to do with the development of good reactor setups for this type of evolutionary experiments. Okay, so that's something that is quite interesting. So in this case, for instance, what we did was to we inserted a pathway for degradation of cellulose. That is one of the residues, one of the components, or one of the components of the treatment of lignocellulosic materials after some manipulations. And then these are good, in principle, these residues are good substrates for a variety of biotechnological applications. And Pseudomonas putida KT2440, I mean, the strain that we use in the laboratory is unable to grow on cellulose. However, what we did was to insert into the chromosome of this bacterium by using a transposon, with the technologies that we discussed yesterday, a pathway for degradation of this sugar. But when we put this pathway in the chromosome, cells didn't grow. So what you do? Well, what you do is that you subject the cells to a continuous culture with lesser concentrations of glucose every time and higher concentrations of cellulose every time. So at the end of the process, at the end of a long evolutionary experiment, you don't have glucose any longer, and what you have is only cellulose as the only carbon source. And then you can select clones that grow, and you look at the chromosome, and you find some mutations here and there. And these mutations obviously represent the result of this heterotic computation that the bacteria have been making, through which bacteria diversify, and then after diversification, then you select for a trait, and that trait is optimal, and the optimality has been obtained not through direct engineering, but through these evolutionary approaches. Okay? It is curious, for instance, that when you look at the mutations that enable this bacterium to grow in this carbon source, you find mutations not only in the pathway itself, but also in other locations here, for instance, in the RPOC gene, that is one of the components of RNA polymerase, okay, indicating that at some point many other genes had to be modified and had to be expressed more or less to ensure that the new pathway could be accommodated in the pre-existing network of the cells. Okay, so I think we should make a little 5 minutes break here, as we do other times, and then we'll continue with more tricks to explore a solution space, as I told you so far. Okay, 5 minutes. What time is it? Okay, be here at 3:10, please. Okay.